So this is the next lesson about the circulatory system, and we're going to be focusing on the blood. So first of all, we need to ask what blood is actually made up of. And as we can see, there are 4 constituents here that you need to know about, and they are the red blood cells, the white blood cells, the plasma and the platelets. And we're going to look at each of these in turn in this lesson. If you have a quick look at the diagram on the right, then you can see that just over half of the blood is the plasma, and the other half is the constituents, the red, white blood cells and the platelets, which you'll find floating around in the plasma. So we're going to make a start by looking at the plasma. Now this is a straw-coloured liquid. It's easy to assume that blood is red because of the red blood cells, but actually the liquid that they float around in is straw-coloured. The plasma transports substances around the body, and we're going to have a look at those in a bit more detail in a moment. But it also distributes heat around the body, which is something we quite often forget about. Again, we look at the movement of heat around the body in a little bit more detail in a unit next year. So what does the blood transport around the body? We know that the circulatory system exists in order to take substances around the body. The first one then is oxygen, which goes from the lungs to all of the cells. Now we looked at that last lesson when we talked about the double circulatory system. We also mentioned carbon dioxide, which is a waste product of respiration that goes from all of the cells and back to the lungs. Glucose then we know we also need for respiration. That comes from the digestive system, along with other food products. Glucose is the one we tend to look at in most detail because of respiration, but all the other food products will also come out of the digestive system and then into the bloodstream. Now that goes to the liver first and then to all of the rest of the cells. Urea then, another waste product, comes from the liver to the kidneys, and we look at urea in a bit more detail again in another unit. But if we're breaking down protein, then part of the waste product is urea. We need to get rid of it, so it's broken down in the liver and then sent to the kidneys. We also transport antibodies around the body. They're produced by white blood cells and they help us fight disease. And we also transport hormones in the blood. They're produced in various glands in the body and then transported to where they're needed by the blood. It is important to point out that oxygen is transported in red blood cells. And again, we'll come to those in a moment. But everything else is transported in the blood plasma. So carbon dioxide, glucose and other food products, urea, antibodies, hormones, all transported by blood plasma. So I'm just going to pause for a moment to recap diffusion and molecule exchange. Now we did meet these last lesson when we talked about gas exchange, but we need to have a slightly wider understanding of this when we're looking at all those different things which are transported in the blood. So if you remember, diffusion is the movement of molecules from a region of high to low concentration. A molecule exchange then is where molecules are transferred or exchanged between the blood and various tissues. And this happens in the capillaries because the capillaries, remember, have really thin walls so that substances can diffuse across them. So as we mentioned last lesson then, oxygen needs to go from the blood into all the cells of the body for respiration. So here it diffuses from the blood in the capillary across that capillary wall, initially into tissue fluid, and from that fluid into the cells, so from a high to a low concentration of oxygen. Carbon dioxide then is going the other way. It's produced in the cells as a waste product of respiration. It diffuses initially from the cell into the tissue fluid, and from the tissue fluid it passes across the capillary wall and into the blood into the capillary. Like oxygen, glucose is needed for respiration, so it's going to move from the blood and into the cells. So again, it's going to diffuse across that capillary wall and into the tissue fluid and into the cells for respiration. Like carbon dioxide, urea is a waste product, so it needs to come from the cells and into the blood. And if you remember, this happens in the liver. So the urea diffuses from the liver cells into the liver tissue fluid, and from the liver tissue fluid across that wall of the capillary and into the blood, or it's going to be carried away by the plasma. So the next part of the blood we're going to have a look at are the red blood cells, and I think we all know that they transport oxygen around the body. For red blood cells to be effective, they need to be able to carry the maximum amount of oxygen and also be able to get through those tiny capillaries, and so they're very specially adapted cells. The first adaptation is having no nucleus. This maximises the amount of space inside the cell for carrying oxygen. The second one is that by being small and flexible, they can fit down through those tiny capillaries. The third is the special shape they have, and if you have a look at the diagram on the bottom right, you'll see they look a little bit like squished in cushions. This shape is called biconcave, where bi means two and concave means sort of scalped in like a cave or pushed in like a cave. So they're sort of squidged in on both sides, biconcave. This gives them a really large surface area for oxygen either to pass in from the lungs or out when it's going to the rest of the cells. By being very thin, it also means the oxygen has the shortest possible distance, again, if it has to diffuse into the cell or out. Red blood cells contain molecules called hemoglobin. Now that HAE at the beginning of hemo means or refers to blood. So in hospital, for example, the hematology department would look at the study of blood and blood tissues. It also refers to iron. So hematite is an iron ore. It's a rock that contains iron oxide. And there's a clear link here because hemoglobin contains iron. In fact, you can see the little diagram on the bottom right there. It's got 4 areas of iron there, and oxygen bonds to that iron, forming something called oxyhemoglobin. So when we breathe oxygen in at the lungs, it goes into the bloodstream and it bonds onto the hemoglobin in red blood cells, forming oxyhemoglobin. So at the lungs we have hemoglobin plus oxygen gives us oxyhemoglobin. When oxygen reaches the cells and needs to be released, then the oxyhemoglobin turns back into hemoglobin plus oxygen, and the oxygen then diffuses into the cells. So the next part of the blood we're going to look at are the white blood cells. They're involved with fighting disease. They ingest pathogens, and remember, pathogens are microbes that can cause us harm, and they produce antibodies. There are several types of white blood cell, and you need to know about 2 of them, and they are the most common ones. The first set are the phagocytes, and they make up about 70% of all the white blood cells. They're the one that get the job of ingesting pathogens. So they engulf and destroy unwanted microorganisms or pathogens by a process called phagocytosis. The picture you can see here on the right sort of shows this. So they detect the thing they want to get rid of, the pathogen. The cell will then surround that pathogen and engulf it, so the pathogen is then pulled into the cell and then it destroys it from within. The other type of white blood cell you need to know about are the lymphocytes, and they make up about 1/4 or 25% of the white blood cells. Now they are the ones that produce the antibodies, and antibodies are soluble proteins, so they dissolve into the blood plasma and then are taken to wherever they're needed in the body. The final part of the blood that you need to know about are the platelets, and they're involved in clotting. They're cell fragments and they're produced by giant cells in the bone marrow. If our bodies get damaged in any way, there is the risk that we could be bleeding, and obviously that's not good. We need our blood to remain inside our body. So platelets are there to stop bleeding, and there are 2 ways that they can do this. The first is that their surface has proteins on that makes them sticky, so they can stick to breaks in a blood vessel wall and they can plug that gap. The second thing they can do is secrete proteins, and these cause further chemical reactions that make blood clot. And when it clots, obviously, it can then plug the gap. So if you cut yourself, then a blood clot will form, and that will then give the skin and the blood vessels and anything else a chance to heal. So basically, it's plugging the gap until the body has repaired itself. A blood transfusion is where we give somebody else's blood to a patient. Surprisingly, these have been going on since the 17th century, but today we don't tend to give whole blood, rather we tend to just give the particular blood product that the patient needs. So that could be red blood cells or platelets or plasma or antibodies. So today the NHS relies on blood donors to give blood, which we then store until it's needed. Obviously we have to check that blood because we don't know whether the donor has been ill, and we screen them for things like infections such as HIV or hepatitis. We check the blood group because you need to match up the blood group of the donor to the patient. That can be really important because if you have a rare blood group, then it might be difficult to get hold of the right blood to transfuse into you. We also screen for the presence of certain antibodies because that might indicate that the donor has had a particular disease. Interestingly, whilst blood transfusions have been around since the 17th century in one form or another, blood banks have only existed since the 1940s. That's all down to the work of this man. This is Charles Drew, known as the father of blood banks. He pioneered the collection, testing and storage of blood plasma, which could then be transfused, and he organised the first large-scale blood bank in the United States. Another project he was involved with was heading up Blood for Britain in World War II. Blood was taken in New York, tested, stored and shipped overseas, obviously saving thousands of British lives. He followed that up by organising a blood bank for the American Red Cross. However, in those days segregation still occurred in the United States and blood products were also segregated. So depending on your race, it would depend on whether or not you could have access to particular blood products. As a prominent African American, Charles Drew spoke out against this, and eventually he resigned his post in protest. But without his pioneering work in the 20th century, thousands, millions of lives would have been lost, so we have an awful lot to thank him for.